Hello everyone, welcome to the new session of Biotechnology in Agriculture Part 1. So in this session, we are going to study about the use of biotechnology in the agricultural field. So let us talk about first that genetically modified crops. What are genetically modified crops? So GM crops or the genetically modified crops or the biotech crops are the plants used in agriculture, the DNA of which has been modified. There is a sort of modification in the DNA of that particular crops, and that is why it is called as biotech crops. Okay, and why it is called as the genetically modified crops? Because its DNA has been modified. Okay, so DNA of which has been modified with the genetic engineering technology, okay, or the genetic engineering techniques. So this is a technology which is used to manipulate or modify the DNA of a particular organism. Okay, so this is a technique. So this technique is used to modify the crops. In most cases, the aim is to introduce a new trait, okay, to the plant which does not occur naturally in the species. So trait, where the traits are present on the gene. Okay, so gene are the responsible for a particular trait. If a gene has been introduced in an species or an organism that corresponds to a particular trait, so that trait will be introduced into that organism. Example in food crops include the resistance to the certain pests. So there are some of the modified crops, and why these food crops has been modified? Because it has to introduce a resistance to certain pests, to certain disease, stressful environment conditions, resistance to the chemical treatment, reduction to the spoilage, or improving the nutrient profile of the crop. So for these so many reasons, the food crops has been modified. Okay, they are examples in non-food crops. Some of the non-food crops are also modified. Why they has been modified? Because include production of pharmaceutical agents, biofuels, and other industrially useful goods, as well as for the bioremediation. For these many reasons, some non-food crops has been modified. Okay, plants and the crops with the GM trait means genetically modified traits have been tested more than any other crops with no credible evidence to harm humans or animals. So there must be a certification that these genetically modified crops are not going to harm in any way to the humans, animals, or even to the environment. So there must be a certification, right? So in fact, seeds with the genetically modified traits have been tested more than any other crops in the history of agriculture. Okay, so with no credible evidence to harm to humans or animals. So the seeds are tested in more than any other crops, okay, in the history of agriculture. So why this has been done? So that they should not harm to animals or humans or even to our environment. Governmental regulatory agencies. Now we are talking about so many agencies that certified that these GM crops are not going to harm in any ways. So the governmental regulatory agencies, scientific organizations, and the leading health association worldwide agreed that food grown from the GM crops is safe to eat. So we can eat these genetically modified crops because they are not going to harm in any ways. The World Health Organization, WHO, American Medical Association, US National Academy of Science, the British Royal Society, among others that have examined the evidence, and all came to the same conclusion that these particular GM crops are not going to harm human, animals, as well as the environment. So whatever we have discussed in the last slide, that there were so many organizations, okay, that concluded that consuming foods containing ingredients derived from the GM crops are not going to harm, and they are safe, and there is no risk in consuming those food or those crop plants, okay, and that genetically modified crops are safe to eat, and there is no risk of consuming those food containing ingredients. So these organizations certified this thing. Genetically modified crops must have made crops more tolerant to the biotic stress. Why these crops has been modified? Because we want a crop that can tolerate the abiotic stress. Abiotic stress means cold, drought, salt, and heat. So we can grow genetically modified crops in drought area, highly salt rich area, the area which is having lots of heat, and cold even. So we have developed so many varieties of the genetically modified crops that can tolerate to the abiotic stress. Reduce reliance on the chemical pesticides. So pest resistant crops has also been introduced. So no need of using those chemical pesticide, as they can harm in some other ways. So pest resistant varieties crops are better to use in comparison to using the chemical pesticides for the conventional crops which were using earlier. Okay, so better to use the GM crops that are having the pest resistance. Help to reduce the post-harvest losses and enhance the nutritional value of the food. So there are many GM crops that has increased the nutritional value of the food. Increased efficiency of the mineral usage by the plants. This prevents early exhaustion of the fertility of the soil. So we have also introduced this technology, has also introduced efficiency, better efficiency of the mineral uses by the plant from the soil, so that fertility of the soil should not be get exhausted, and adequate amount of minerals from the soil can be used. So it prevents the exhaustion of the fertility of the soil even. Enhanced nutritional value of food, like example golden rice that is rich in vitamin A. Okay, so this particular rice, golden rice, is rich in vitamin A. So this increases the nutritional value of the rice. This is one of the example. Okay, so there are so many technologies has been introduced in the biotechnology. Okay, so we are going to discuss some of the technology that are used in agriculture. So one of the technique in the biotechnology is the RNA interference. So what is that? RNA interference is a method of blocking gene function by inserting sort of sequences of ribonucleic acid RNA that match part of the target gene sequence. Thus, no proteins are produced. So there is an RNA interference. Because we want to interfere that particular RNA, because this protein synthesized from this particular RNA going to harm in some or the other way to the plants. Okay, so we want to block this RNA. Okay, that is why it is called as RNA interference. So this method is a blocking gene. Because we are blocking RNA means the effect of that particular gene is blocked. Okay, so that is why it is called as RNA interference. We will see how the mechanism of RNA interference work in the coming slide. So RNA has the potential to become a powerful therapeutic approach towards the targeted and the personalized medicine. RNAi has provided a way to control pests and diseases, introduce novel plant trait, and increase crop yield. So this particular technique has been used to control pests or disease in the plants, and by introducing this particular technique in the plant, we have increased the yield of the crop. RNAi take place in all eukaryotic organisms as a method of cellular defense. So this is a method of cellular defense in the eukaryotic organism. This method involves silencing of specific mRNA. So mRNA is silenced, means no production of protein, from this mRNA. Okay, so this is called silencing because we are blocking this protein formation. Okay, so this method involves silencing of the specific mRNA due to the complementary double-stranded RNA molecule that binds to and prevent the translation of the mRNA, that is called as silencing. So double-stranded RNA molecule, when formed, it does not produce any functional protein, and this is called silencing. So let us see the mechanism of this particular RNA interference. The source of this complementary RNA could be formed an infection by viruses having RNA genomes or a mobile genetic elements called transposons that replicate via an RNA intermediate. So what happened? Transposon, let us first understand what are the transposon. Transposon are some sort of a gene that jumps from one position to the another in a genome, and that is why it is also called as the jumping gene. Okay, so what happened? RNA genome or mobile genetic elements replicate via an RNA intermediate. So this RNA replicate means makes its copy. Okay, and what happens after that? Scientists have developed a novel crop such as nicotine-free tobacco, non-allergenic peanuts, decaffeinated coffee, and nutrient-fortified maize from this particular technique. Mechanism of RNA interferences as understood is that it comes into play when a double-stranded RNA is introduced either naturally or artificially in the cell. So this particular RNA interference occur only when there is the synthesis of double-stranded RNA. So how it comes? It is either introduced naturally or artificially in the cells, and scientists have developed so many things with the help of this RNA interference, like nicotine-free tobacco, non-allergenic peanuts, decaffeinated coffee, and nutrient-fortified maize. An endoribonuclease enzyme cleaves the long double-stranded RNA into small piece of RNA. So double-stranded RNA has been synthesized. Now what happened? An endoribonuclease enzyme cleaves the long double-stranded RNA into small piece of RNA. Now these small piece of RNA could be miRNA or siRNA depending upon the origin of the long double-stranded RNA, that is endogenous or exogenous respectively. So this particular double-stranded RNA can be exogenous or endogenous. Okay. So that can be introduced from outside as well as they can be synthesized inside only. So that is why double-stranded can be exogenous or endogenous. Double-stranded RNA may be generated either by RNA-dependent RNA polymerase. So RNA-dependent RNA polymerase means single-stranded RNA. Now there is a RNA dependent on the template of RNA. New RNA is synthesized. So this particular RNA polymerase is RNA dependent RNA polymerase. This particular polymerase enzyme required RNA as a template. So this mRNA is there. On this mRNA, a new RNA is synthesized with the help of RNA dependent RNA polymerase enzyme. Okay, or bidirectional transcription of the transposable elements or physically introduced. Sometimes it is also exogenously introduced. So like this, the double-stranded RNA get synthesized. Okay. Dicer. Now comes this dicer. Dicer, also known as endoribonuclease, encoded by the dicer gene. Now dicer cleaves the double-stranded RNA and pre-microRNA or pre-miRNA into short double-stranded RNA fragments called small interfering RNA or the microRNA. So dicer cleaves this particular double-stranded RNA into small interfering RNA or the microRNA. The RNA-induced silencing complex, that is RISC, here we can see in this diagram, RISC binds to the short domain of RNA, and long double-stranded RNA is cleaved into short double-stranded RNA. So here, this RISC helps to bind to the short domains of RNA and long double-stranded RNA and cleaves into shorter double-stranded RNA fragments. So this dicer and this RISC is a very, very important for cleaving or fragmenting the double-stranded RNA into small fragments of RNA or short double-stranded RNA. There are several opportunities for the applications of RNAi in crops science for its improvements such as stress tolerance and enhanced nutritional level. So this particular technique has been used for improvement of crops in the field of stress tolerance and enhancing the nutritional value of the crop. Okay. So now here you can see this, the double-stranded fragments of the RNA with the help of RISC is cleaved into sliced mRNA. This you can see the sliced mRNA. Now this mRNA is of no use, and that is why it is called as silencing. This knockdown technology may be useful in inducing early flowering, delaying ripening, delaying senescence, breaking dormancy, stress-free plants, overcoming self-fertility. So this particular technology has been introduced so many things in the crops like early flowering, delaying ripening, delaying senescence, breaking the dormancy, stress-free plants, and overcoming the self-sterility. Means they are used for improving the quality of the crop plants, and that is why it is called as genetically modified crops. RNA interference has recently been demonstrated in the plants parasitic nematodes. So this has been introduced in plant parasitic nematodes, the nematodes that are parasites to the plants. So it is a potentially powerful investigative tool for the genome-wide identification of gene function that should help improve our understanding of plant parasitic nematodes. So this particular technique, this parasitic nematode help us to identify that gene that should help or improve our understanding of the plant parasitic nematode. And what is the result of that? This RNA should help identify the gene and hence the protein targets for the nematode. Why this particular nematode is eating that plant? For particular protein, because they want to target a protein, right? And if you do not form that particular protein in the plant, what will happen? This nematode would die. So the RNA should help identify that particular gene or that particular protein that get digested in their intestine. Okay, and we can control the nematode in that way. The prospects for the novel resistance depend on the plant generating an effective form of double-stranded RNA in the absence of an endogenous target gene without determinant to itself. These RNA molecules must then become available to the nematode and be capable of ingesting via its feeding tube. So this particular RNA molecule must then become available to the nematode. This RNA must be available to the nematode and capable of ingesting via its feeding tube. So that particular RNA should be capable of ingesting via feeding tube. If this requirement can be met, crop resistance could be achieved by plant delivering a double-stranded RNA that targets a nematode gene and induces a lethal or highly damaging RNAi effect on the parasites. So what happened? This particular double-stranded RNA target a gene in the nematode and that gene is silenced. And that is a lethal effect in the nematode. And that helps the nematode to die and that we can get rid of that parasite. Next use of biotechnology is delaying with the fruit ripening. So delaying fruit ripening is a process of interest because it increases the shelf life of this fruit. Okay, so by using this technology, we can introduce or increase the shelf life of the fruit as it allows more time for the shipment of fruit from the farmer's field to the grocer. So suppose this is the farmer's field, this is a farmer's field and this is the grocer. Okay, so if the farmer is using this or growing the genetically modified crop, then it has a longer time to do the shipment from its field to the grocer, because this particular fruit or the crop is not going to perish, okay, because the shelf life of the fruit has been increased now by using this particular technique. So if the farmer is using or growing this particular GM crops, okay, then it has an advantage that the longer time of keeping that fruit and transporting it to the grocer. Ethylene is a natural plant hormone. Now, what is the particular science behind this particular GM crop? Ethylene, this is a particular plant hormone or the phytohormone associated with the growth, development and ripening and aging of many plants. So this ethylene causes ripening and aging in the plants. Okay, this we all know. Now, delay with the fruit ripening. How to delay this fruit ripening? Ethylene phytohormone is said to promote the ripening in the variety of fruits, okay, like banana, pineapple, tomatoes, mangoes, melons, and papayas, right? So if we can block the synthesis of this particular ethylene or minimize the synthesis of this ethylene, we have a controlled over the ripening of the fruit, right? So let us see how it happens. The amount of ethylene produced can be controlled primarily by switching off or decreasing the production of ethylene in the fruit, and there are several ways to do this. So how to block the synthesis of ethylene? Synthesis of ethylene blocked, right? How to do this blocking of synthesis of ethylene? Let us see that. So separation, first thing is suppression of ACC synthase gene expression. Now what is ACC? ACC is the amino acyl propane carboxylic acid. This is a compound, okay? Now ACC synthase is an enzyme responsible for the conversion of adenosine methionine, S-adenosine methionine, that is SAM, to ACC, that is amino acyl propane carboxylic acid. So here we are having SAM, right? This SAM, that is S-adenosyl methionine, S-adenosyl methionine, right? So this is a compound. Now it get converted to amino cyclopropane carboxylic acid, right? Now for this we need an enzyme conversion of S-adenosyl methionine to amino acyl cyclopropane carboxylic acid require an enzyme. And what is that enzyme? ACC synthase. ACC synthase. ACC synthase. ACC synthase. Okay. So the second is this is the last step in the ethylene biosynthesis. So now here you can see ACC is converted to ethylene with the help of an enzyme ACO, that is amino cyclopropane oxidase. Okay, so if this ACC is not formed or the synthesis is blocked in any way, right, what will happen? The ethylene synthesis will be affected. So enzyme expression is hindered when an antisense, mirror image, or truncated copy of synthase gene is inserted into the plant genome. Now what we are doing? We are ACC synthase. This is this gene. There is a gene for this. Gene for ACC. Now this gene is manipulated. And its mRNA will also be manipulated. Manipulated means it's not an original ACC gene, or this mRNA is also not the functional mRNA. So if the protein is synthesized, that is that enzyme ACC synthase, this will be truncated copy. It means not the original copy. This is not the original ACC synthase and not going to convert SAM to ACC. This will be affected. Okay. So the enzyme expression is hindered when an antisense or the truncated copy of synthase gene is inserted into the plant's genome. So by the gene manipulation, by the biotechnology technique, we are manipulating the gene of ACC, right? So the gene coding for the enzyme is obtained from the Pseudomonas chlororhabes, a common non-pathogenic soil bacterium. Okay, so we are taking this particular organism to take out a gene. Now it convert ACC to a different compound, thereby reducing the amount of ACC available for the ethylene production. So when this particular gene is introduced, taken out from this particular soil bacterium and introduced in the plant, so this gene result a protein that affect ACC gene, and when this ACC gene is affected, it will produce a truncated mRNA. So it will not produce a functional ACC synthase, right? So this is the technique we are introducing in the crop. We are introducing a particular gene from the Pseudomonas chlororhabes. That protein, this gene will code a protein, right? And this protein is going to affect this ACC gene. So this approach is similar to the ACC deaminase, wherein the ethylene production is hindered when the amount of its precursor metabolite is reduced. In this case, SAM is converted to the homoserine. Now ACC deaminase there is another approach to affect the synthesis of ACC. Okay, SAM is affected in such a way that it convert. Now in the presence of another enzyme, this SAM is converted to a homoserine. The gene coding for the enzyme is obtained from E. coli T3 bacteriophage. So this ACC deaminase enzyme is introduced via a gene that is taken out from the E. coli T3 bacteriophage. So this is another way of silencing the ACC production. ACC oxidase is the enzyme which catalyzes the oxidation of ACC to ethylene, the last step in the ethylene biosynthetic pathway. So here you can see ACC oxidase. This is ACC oxidase or ACO oxidase, right? So this is the final enzyme that is required to convert ACC to ethylene. But we have discussed so many ways to reduce the production of ACC, okay, by affecting the SAM or introducing another enzyme, right? So if this ACS is lower down, the ACC production will be affected. So there will be reduction in the production of ethylene enzyme, and this fruit will not going to ripe. And in this way, we are going to increase the shelf life of the fruit. Through antisense technology, down regulation of the ACC oxidase gene result in the suppression of ethylene production, thereby delaying the fruit ripening. Now, another antisense technology we are using to affect the ACC oxidase gene. If the ACC oxidase gene affected, if there is a lot of production of ACC, then also this ACO is not present, or truncated form or manipulated form. So that is not going to change ACC to ethylene, right? So in this way, we can also suppress the production of ethylene and thereby delaying the fruit ripening. So this is all about this session. We will have some more session on the biotechnology to understand some other examples also, to knowing this particular technology in the better way, how it has been used in the agriculture. Till then, thank you very much.